Chapter 9. The Central Nervous System. This chapter provides a comprehensive overview of the nervous system, focusing on its evolution, structure, and function. We begin by examining the evolution of nervous systems, exploring how neural structures have developed across different species. The anatomy of the central nervous system section details the organization of the brain and spinal cord, highlighting their protective features and functional divisions. The spinal cord is discussed as a critical pathway for communication between the brain and the body. The brain section covers its major regions, including the cerebrum, cerebellum, brainstem, and limbic system, outlining their roles in cognition, movement, and autonomic regulation. Finally, the brain functions section delves into sensory and motor processing, language, memory, and higher cognitive abilities. Together, these topics form the foundation for understanding how the nervous system regulates physiological processes and enables complex behaviors. In vertebrate evolution, the most dramatic changes are seen in the forebrain region. This part of the brain has undergone significant expansion, particularly in more advanced species. The forebrain includes the cerebrum, which plays a crucial role in higher cognitive functions, sensory processing, and voluntary motor control. The nervous system has undergone significant evolutionary changes across different species. This figure illustrates the step-by-step progression from simple nerve nets to the highly developed human brain. Panel. The most basic form of a nervous system is seen in jellyfish, which have a nerve net instead of a centralized brain. Panel B. In flatworms, we see the development of a primitive brain and a nerve cord, marking an important step towards centralization. Panel C. Roundworms further evolve a single brain and multiple ganglia, which help process information locally. Panel D. In fish, the forebrain begins to expand, signaling early advancements in sensory processing and coordination. Panel E. As we move to birds, the cerebellum and forebrain become larger, supporting more complex behaviors like flight and spatial navigation. Panel F. Finally, in humans, the forebrain dominates brain structure, allowing for higher cognitive functions, problem solving, and advanced communication. This evolutionary trajectory highlights the increasing complexity and specialization of the nervous system, shaping the behavioral and cognitive abilities of different species. The central nervous system, or CNS, develops from a hollow tube during early embryonic development. It begins as a specialized group of cells called the neural plate, which serves as the foundation for the brain and spinal cord. Around day 23 of development, the neural plate fuses to form a neural tube. The most caudal or inferior part of this tube will later develop into the spinal cord. The hollow center, or lumen, of the neural tube does not disappear. Instead, it remains as the ventricular system of the brain, which is responsible for producing and circulating cerebrospinal fluid. This early developmental process is crucial for forming the structure of the CNS and ensuring proper nervous system function later in life. The formation of the human nervous system begins early in embryonic development. This figure highlights two crucial stages, day 20 and day 23. On day 20, in a dorsal view of the embryo, specialized cells from the neural plate migrate toward the midline. Accompanying them are neural crest cells, which will later contribute to the formation of structures in the peripheral nervous system. By day 23, the formation of the neural tube is nearly complete. The anterior and posterior openings of the neural tube remain temporarily open before fully closing. The neural crest cells will differentiate into structures of the peripheral nervous system, while the neural tube itself will develop into the central nervous system, forming the brain and spinal cord. This process is a fundamental step in nervous system development, ensuring the proper formation and specialization of the brain and spinal cord. As the human nervous system continues to develop, significant structural changes occur from birth through childhood. At 40 weeks, which is full-term birth, the cerebrum has expanded and now covers most of the other brain regions. The rapid growth of the brain within the confined space of the skull causes it to develop gyri and sulci, forming the characteristic folds on the surface of the brain. These folds increase surface area, allowing for greater neural connectivity and processing power. During childhood, additional structural and functional changes continue. The orientation of the nervous system changes due to the natural flexion of the neural tube during development. The terms dorsal and ventral, meaning upper and lower, are used differently in the brain compared to the spinal cord. The rostral and caudal directions, referring to the front and back, also shift as the brain matures. These adaptations in nervous system development allow for increasing cognitive function, motor control, and sensory processing as a child grows. This figure illustrates the posterior view of the central nervous system, highlighting the spinal cord and its connections to the brain. The spinal cord serves as the main communication highway between the brain and the rest of the body. It is protected by the vertebral column and is organized into 31 pairs of spinal nerves, which exit the spinal column at different levels. There are 8 cervical nerves located in the neck region. Moving downward, 12 thoracic nerves are found in the upper back. Below them, 5 lumbar nerves extend into the lower back. Further down, 5 sacral nerves are positioned near the pelvis. Finally, at the base of the spinal column, 1 coccygeal nerve is present. These spinal nerves allow for motor and sensory communication, ensuring proper function in movement, reflexes, and sensation throughout the body. The spinal cord plays a crucial role in transmitting signals between the brain and peripheral nervous system, making it an essential component of the central nervous system. The central nervous system consists of the brain and the spinal cord. Within the central nervous system, there are 2 distinct types of tissue: gray matter and white matter. Gray matter is composed primarily of nerve cell bodies, which are responsible for processing and integrating information. It also contains clusters of cell bodies in the central nervous system known as nuclei, along with dendrites, axon terminals, and unmyelinated axons. These structures play a crucial role in communication within the nervous system by transmitting and processing signals. White matter, in contrast, consists mostly of myelinated axons. Myelin is a fatty substance that surrounds axons, allowing for faster signal transmission. Within white matter, axon bundles connect different regions of the central nervous system, forming pathways known as tracts. These tracts facilitate communication between various parts of the brain and between the brain and spinal cord. Together, gray and white matter work in coordination to ensure proper nervous system function, allowing for complex processing, sensory interpretation, and motor control. This figure presents a posterior view of the spinal cord along with its surrounding structures. The spinal cord is housed within the vertebral column, which provides structural support and protection. The spinal cord consists of both gray matter and white matter. The gray matter, located in the center of the spinal cord, contains nerve cell bodies and is responsible for processing information. Surrounding the gray matter is the white matter, which consists of myelinated axons that facilitate rapid communication between different regions of the nervous system. The central canal runs through the center of the spinal cord and contains cerebrospinal fluid, which helps cushion and nourish nervous tissue. Extending from the spinal cord are spinal nerves, which connect the central nervous system to the rest of the body, allowing for motor control and sensory input. Surrounding the spinal cord are three protective layers of connective tissue known as the meninges. The innermost layer, called the pia mater, is a thin and delicate membrane that directly covers the spinal cord. The arachnoid membrane is the middle layer, providing additional cushioning and structural support. The outermost layer, the dura mater, is a thick and durable membrane that offers the greatest level of protection. Also visible in this diagram are the autonomic ganglia, which are clusters of nerve cells that play a role in regulating involuntary functions such as heart rate, digestion, and respiration. Together, these structures ensure the proper function and protection of the spinal cord, allowing it to serve as the main communication pathway between the brain and the body. The brain is encased in a protective bony structure known as the cranium, which serves to shield it from physical injury. Similarly, the spinal cord is housed within the vertebral column, providing structural support and safeguarding the delicate nervous tissue from mechanical damage. To further protect the brain and spinal cord, connective tissue layers known as the meninges lie between the bone and neural tissue. These layers serve to stabilize the nervous system structures and prevent injury from movement or impact. There are three primary meningeal layers, listed from the deepest to the most superficial. The pia mater is the innermost layer, adhering closely to the surface of the brain and spinal cord. It is a thin, delicate membrane that contains blood vessels to nourish the nervous tissue. Above this lies the arachnoid membrane, a web-like structure that acts as a cushion, allowing cerebrospinal fluid to circulate and absorb shock. The outermost layer is the dura mater, a tough, durable membrane that provides the greatest degree of protection, forming a strong barrier between the brain and the skull or between the spinal cord and the vertebral column. A helpful way to remember the order of these layers from deep to superficial is with acronym PAD, representing the pia mater, arachnoid membrane, and dura mater. These structures work together to maintain the integrity and stability of the central nervous system, preventing damage and ensuring optimal function. This figure provides a sectional view of the meninges, which are the protective membranes that cushion and support the brain. These layers, along with extracellular fluid, play a crucial role in protecting the delicate brain tissue from injury. At the outermost level, the cranium encases the brain, providing a rigid structure that shields it from external impact. Just beneath the cranium lies the dura mater, which is the thickest and most durable of the meningeal layers. It consists of two sublayers, the periosteal layer, which is attached to the inner surface of the skull, and the meningeal layer, which forms the true protective covering around the brain. Beneath the dura mater is the arachnoid membrane, a delicate, web-like structure. This membrane encloses the subarachnoid space, which serves as a critical area for cushioning the brain. Within this space, cerebrospinal fluid, or CSF, circulates, providing both physical protection and nutrient transport to neural tissues. The pia mater is the innermost meningeal layer, lying directly on the surface of the brain. This thin membrane contains numerous blood vessels that supply oxygen and nutrients to the brain tissue. Additionally, the venous sinus is visible in this diagram. It serves as a drainage system for cerebrospinal fluid and venous blood, helping maintain intracranial pressure and fluid balance within the brain. Together, the meninges, cerebrospinal fluid, and venous drainage system work to protect, support, and maintain the overall function of the central nervous system. The brain and spinal cord are surrounded and cushioned by cerebrospinal fluid, a salty solution similar in composition to plasma. This fluid serves essential functions in protecting and maintaining the central nervous system. Cerebrospinal fluid is produced by the choroid plexuses, which are networks of capillaries located in the ventricles of the brain. Within these capillaries, certain materials, including sodium and other solutes, are selectively moved from the plasma into the ventricles. This process creates an osmotic gradient, drawing water into the ventricles and ensuring a continuous flow of cerebrospinal fluid. Once produced, cerebrospinal fluid circulates around the entire brain and spinal cord. It is contained within the subarachnoid space, which lies between the arachnoid membrane and the pia mater. The fluid flows from the ventricles into the subarachnoid space and eventually returns to the plasma through specialized structures called arachnoid villi. Cerebrospinal fluid performs several critical functions. It provides physical protection by cushioning the brain against impacts and sudden movements. It also reduces the effective weight of the brain, preventing excessive pressure on the delicate nervous tissue. Additionally, cerebrospinal fluid plays a role in nutrient and waste exchange, allowing for the transport of essential molecules and the removal of metabolic byproducts from the central nervous system. This fluid environment is vital for maintaining the stability and function of the brain and spinal cord, ensuring optimal conditions for neural activity and communication. This figure illustrates the ventricles of the brain, which are fluid-filled cavities that play a crucial role in the production and circulation of cerebrospinal fluid. The ventricular system consists of interconnected chambers that allow cerebrospinal fluid to flow through the central nervous system. The first and second ventricles, known as the lateral ventricles, are the largest and are located within each hemisphere of the brain. These ventricles connect to the third ventricle through small openings known as the interventricular foramina. The third ventricle is a narrow cavity situated in the midline of the brain, within the diencephalon. From the third ventricle, cerebrospinal fluid flows downward through the cerebral aqueduct, a slender canal that connects the third ventricle to the fourth ventricle. The fourth ventricle is located between the brainstem and the cerebellum. As the fluid exits the fourth ventricle, it continues downward into the central canal of the spinal cord, ensuring continuous circulation throughout the central nervous system. The ventricular system not only provides a pathway for cerebrospinal fluid circulation, but also helps cushion the brain, regulate pressure, and remove metabolic waste. This interconnected system is essential for maintaining the stability and function of the brain and spinal cord. Cerebrospinal fluid is secreted into the ventricles of the brain, where it circulates through the subarachnoid space. This fluid plays a critical role in cushioning the central nervous system, protecting it from mechanical shock and maintaining a stable chemical environment for neural function. The choroid plexus is the specialized structure responsible for cerebrospinal fluid production. It is located within the ventricles and consists of a network of capillaries surrounded by specialized ependymal cells. These cells actively transport water, ions, and nutrients from the blood into the cerebrospinal fluid, ensuring that the nervous system receives the necessary components for proper function. Once secreted, cerebrospinal fluid flows through the ventricular system, circulates around the brain and spinal cord, and is eventually reabsorbed into the bloodstream through the arachnoid villi. This continuous cycle helps regulate intracranial pressure and maintain homeostasis within the central nervous system. The production and circulation of cerebrospinal fluid are essential for supporting brain function, providing both physical protection and metabolic support to the neurons of the central nervous system. Cerebrospinal fluid is continuously produced and circulated within the central nervous system, but it must also be reabsorbed to maintain proper balance and intracranial pressure. This figure illustrates how cerebrospinal fluid is reabsorbed into the bloodstream. The process of reabsorption occurs at specialized structures called arachnoid villi, which are small, finger-like projections of the arachnoid membrane. These villi extend into the venous sinuses, large blood-filled spaces located within the dura mater. As cerebrospinal fluid flows through the subarachnoid space, it moves into the arachnoid villi, where it is transferred into the venous blood. The endothelial lining of the venous sinus facilitates this movement, allowing excess cerebrospinal fluid to drain into the bloodstream. This process ensures that the volume and composition of cerebrospinal fluid remain stable, preventing the accumulation of excess fluid, which could lead to increased intracranial pressure. The continuous cycle of secretion, circulation, and reabsorption of cerebrospinal fluid plays a crucial role in maintaining the chemical environment of the brain, providing protection and facilitating nutrient and waste exchange within the central nervous system. The blood-brain barrier is a highly specialized structure that regulates the movement of substances between the bloodstream and the brain. This barrier is formed by the tight junctions between endothelial cells of brain capillaries, which limit the passage of harmful substances while allowing essential nutrients to reach brain tissue. Astrocytes, a type of glial cell, play a crucial role in maintaining the blood-brain barrier. These cells secrete chemical signals that reinforce tight junctions, ensuring that the barrier remains intact. By doing so, astrocytes help protect the brain from toxic water-soluble compounds and pathogens, preventing harmful substances from entering neural tissue. Although the blood-brain barrier is highly selective, certain molecules can cross it. Small lipid-soluble molecules, such as oxygen, carbon dioxide, and ketones, diffuse freely across the barrier. However, larger hydrophilic molecules, including glucose and amino acids, require specialized transport proteins to move into the brain. Additionally, parasites are another key component of the blood-brain barrier. These specialized cells are located between endothelial cells and the foot processes of astrocytes. Parasites secrete paracrine signals, which help regulate and maintain the tight junctions, further reinforcing the barrier's protective function. This selective permeability of the blood-brain barrier ensures that the brain remains protected from toxins while still receiving the necessary nutrients for normal function. In the top image, a cerebral angiogram reveals the extensive blood supply to the brain. The brain contains approximately 400 miles of capillaries, ensuring that oxygen and nutrients are efficiently delivered to neural tissue. Major arteries, including the anterior cerebral artery, posterior cerebral artery, middle cerebral artery, internal carotid artery, and the circle of Willis, play a key role in distributing blood throughout the brain. Despite this vast network of blood vessels, neurons require protection from potentially harmful substances in the bloodstream. Unlike typical capillaries, brain capillaries are not leaky. The tight junctions between endothelial cells form a selective barrier that prevents harmful substances from diffusing into the brain while allowing essential nutrients to pass. Astrocytes, a type of glial cell, contribute to the maintenance of the blood-brain barrier. The foot processes of astrocytes release chemical signals called paracrines, which promote the formation and reinforcement of tight junctions between endothelial cells. This helps maintain the integrity of the barrier, ensuring that only select molecules can pass into the brain. Additionally, parasites, which are located along the walls of brain capillaries, provide structural support and regulate blood flow. These specialized cells work alongside astrocytes to further strengthen the blood-brain barrier and modulate its permeability. Together, these features of the blood-brain barrier ensure that neurons are protected from toxins and fluctuations in blood composition, maintaining the brain's stable internal environment for optimal function. Neurons require a constant and uninterrupted supply of oxygen and glucose to function properly. The brain is one of the most metabolically active organs in the body and demands a significant amount of energy to sustain its activity. To meet these energy needs, the brain receives approximately 15% of the blood pumped by the heart. This ensures a steady supply of essential nutrients to support neuronal activity and maintain cognitive function. Oxygen is a crucial component of cellular respiration, allowing neurons to generate the energy required for neurotransmission. It freely crosses the blood-brain barrier, diffusing directly from the bloodstream into brain tissue without the need for specialized transport mechanisms. Glucose is another essential fuel source for the brain. Unlike oxygen, glucose requires membrane transporters to move from the plasma into the brain's interstitial fluid. This process is facilitated by GLUT1 transporters, which allow glucose to enter neurons without requiring insulin. The brain is responsible for nearly half of the body's total glucose consumption, highlighting its high metabolic demand. Since neurons have very limited energy storage, they rely on a continuous supply of glucose from the bloodstream. Any disruption in glucose availability, such as in cases of hypoglycemia, can impair cognitive function, leading to confusion, dizziness, and even loss of consciousness. This constant need for oxygen and glucose underlines the importance of maintaining adequate blood flow to the brain, ensuring that neurons receive the energy they require to sustain thought, movement, and overall brain function. The spinal cord is a vital structure within the central nervous system, serving as the primary communication pathway between the brain and the rest of the body. It is organized into 31 pairs of spinal nerves, which branch from the spinal cord to innervate different regions of the body. Each spinal nerve exits the spinal cord through the spaces between the vertebrae and immediately branches into two roots, the dorsal root and the ventral root. The dorsal root is responsible for carrying sensory information into the central nervous system. Sensory neurons transmit signals from the body to the spinal cord, where they synapse onto interneurons in the dorsal horn of the spinal cord. Located within the dorsal root is the dorsal root ganglion, which contains the cell bodies of afferent sensory neurons. This structure plays a key role in processing and relaying sensory information to higher brain centers. In contrast, the ventral root carries motor information from the central nervous system to the muscles and glands. The cell bodies of motor neurons are located in the ventral horn of the spinal cord. These neurons generate motor commands that control voluntary and involuntary movements, allowing for coordinated muscle activity and reflex responses. The interaction between the dorsal and ventral roots enables efficient communication between the central nervous system and the peripheral nervous system. This system ensures that sensory inputs are processed appropriately and that motor outputs are executed effectively to maintain movement, posture, and reflexive actions. The spinal cord consists of gray matter and white matter, each playing a distinct role in nervous system function. Gray matter is organized into 3 horns, each with specific functions. The dorsal horns contain visceral and somatic sensory nuclei. Visceral sensory nuclei process signals from internal organs, while somatic sensory nuclei receive input from the skin, muscles, joints, tendons, and ligaments. The lateral horns contain visceral motor nuclei that control autonomic functions, regulating smooth muscle, cardiac muscle, and glands. These autonomic motor neurons play a key role in involuntary functions such as digestion and heart rate regulation. The ventral horns house somatic motor nuclei, which send signals to skeletal muscles to coordinate voluntary movement. These motor neurons control muscle contractions essential for posture, movement, and reflex responses. White matter surrounds the gray matter and is divided into columns and tracts, composed mainly of myelinated axons. These axons form pathways for communication between different parts of the nervous system. There are two main types of tracts within the white matter. Ascending tracts, which are afferent pathways, carry sensory information from the body to the brain. In contrast, descending tracts, which are efferent pathways, transmit motor commands from the brain to the spinal cord, allowing for movement and reflex control. Additionally, the spinal cord plays a crucial role in integrating spinal reflexes, such as the patellar tendon reflex, which allows for rapid, involuntary responses to stimuli. These reflexes are processed at the level of the spinal cord without requiring input from the brain, ensuring quick and protective reactions to sudden changes in the environment. This structural organization of the spinal cord enables efficient sensory processing, motor control, and reflex integration, making it a critical component of the central nervous system. This figure illustrates the organization of the spinal cord, showing the relationship between gray matter, white matter, and spinal nerve pathways. In the first diagram, a ventral view of a spinal cord segment displays one pair of spinal nerves exiting the vertebral column. Each spinal nerve splits into two roots. The dorsal root, which carries sensory, afferent information into the central nervous system, and the ventral root, which carries motor, efferent commands to muscles and glands. The second diagram highlights the composition of gray matter within the spinal cord. The dorsal horn contains sensory nuclei, which are further divided into visceral sensory nuclei, responsible for processing signals from internal organs, and somatic sensory nuclei, which receive input from the skin, muscles, and joints. The lateral horn houses autonomic efferent nuclei, which control involuntary motor functions, including those regulating smooth muscle, cardiac muscle, and glandular activity. The ventral horn contains somatic motor nuclei, responsible for transmitting motor commands to skeletal muscles, enabling voluntary movement. The third diagram focuses on white matter, which consists of tracts of axons that relay signals between the brain and the spinal cord. There are two primary types of tracts within white matter. Ascending tracts, shown in green, carry sensory information from the spinal cord to the brain. Descending tracts, shown in blue, transmit motor commands from the brain to the spinal cord, where they activate appropriate responses. This structured organization allows the spinal cord to efficiently process sensory input, motor output, and reflex integration, making it an essential component of the central nervous system. This figure illustrates the mechanism of spinal reflexes, which are rapid, involuntary responses to stimuli that occur without direct input from the brain. A reflex arc begins when a stimulus is detected by sensory receptors. This sensory information is transmitted via afferent neurons to the spinal cord, where it is processed by an integrating center. The integrating center, located within the spinal cord, consists of interneurons that connect sensory input with motor output. Once processed, a command is sent to muscles or glands via efferent neurons, triggering an immediate response. Because the spinal cord can generate a reflexive action independently, This allows for faster reactions to potentially harmful stimuli, such as pulling away from a hot surface or quickly adjusting posture to maintain balance. Although the reflex occurs without input from the brain, sensory information about the stimulus may still be relayed to higher brain centers for further processing. This allows the brain to become aware of the event and possibly modify future reflex responses. Spinal reflexes play a crucial role in maintaining posture, balance, and protective responses, ensuring rapid adjustments to environmental changes without requiring conscious thought. The brain is the control center of the body, coordinating sensory input, motor output, and autonomic functions. One key aspect of brain function involves cranial nerves, which serve as direct communication pathways between the brain and various regions of the head, neck, and internal organs. Out of the 12 cranial nerves, 10 originate from the brainstem, making this region essential for basic physiological functions. Cranial nerves can contain sensory fibers, which transmit afferent signals, somatic motor fibers, which control voluntary movements, and autonomic fibers, which regulate involuntary processes. Some cranial nerves contain a mixture of these fiber types, making them functionally diverse. One important example is the vagus nerve, also known as cranial nerve 10. The vagus nerve is classified as a mixed nerve, meaning it carries both sensory and motor fibers. Additionally, it is a key component of the parasympathetic nervous system, containing approximately 75% of all parasympathetic fibers in the body. This allows it to regulate critical autonomic functions such as heart rate, digestion, and respiratory activity. The brainstem also contains the reticular formation, a complex network of neurons that extends throughout the brainstem and plays a fundamental role in regulating wakefulness, sleep, muscle tone, and pain modulation. The reticular formation also houses nuclei that control essential autonomic functions, including heart function, blood pressure, and respiration. Together, the cranial nerves, brainstem, and reticular formation ensure that the brain effectively processes sensory information, initiates motor commands, and maintains homeostasis, keeping the body functioning efficiently. This table summarizes the 12 cranial nerves, classified as sensory, motor, or mixed based on their function. The olfactory and optic nerves are purely sensory, responsible for smell and vision, respectively. The oculomotor, trochlear, and abducens nerves control eye movements, while the trigeminal nerve manages facial sensation and chewing. The facial nerve controls facial expressions and taste, while the vestibulocochlear nerve regulates hearing and balance. The glossopharyngeal nerve aids in swallowing and salivation, and the vagus nerve plays a key role in autonomic functions like heart rate and digestion. The spinal accessory nerve controls neck and shoulder movements, and the hypoglossal nerve governs tongue movement for speech and swallowing. The brainstem is a critical structure that connects the brain to the spinal cord and regulates essential involuntary functions. It consists of three main regions: the medulla oblongata, pons, and midbrain. The medulla oblongata contains both sensory and motor pathways. Somatosensory tracts transmit sensory signals, while corticospinal tracts carry motor commands to skeletal muscles. Within the medulla, pyramidal tracts facilitate voluntary movement, and approximately 90% of corticospinal fibers cross to the opposite side of the body. The reticular formation within the medulla controls vital autonomic functions, including blood pressure, breathing, swallowing, vomiting, and cardiac regulation. The pons acts as a relay station between the cerebrum and cerebellum, facilitating communication between motor areas to coordinate movement. It also plays a role in breathing regulation, working in conjunction with the reticular formation to adjust respiratory rhythms. The midbrain is responsible for eye movement and reflexive responses to auditory and visual stimuli. It serves as a relay center for hearing and vision reflexes, allowing rapid adjustments to environmental changes. Together, these three regions of the brainstem ensure that the body maintains essential autonomic functions while integrating sensory and motor information for coordinated movement and reflexes. This figure provides a lateral view of the brainstem, highlighting its structural components and essential functions. At the top of the brainstem, the midbrain plays a role in eye movement and reflexive responses to visual and auditory stimuli. The pons, located below the midbrain, acts as a relay station between the cerebrum and cerebellum, allowing for coordinated motor control. It also assists in the regulation of breathing rhythms by communicating with respiratory centers. The medulla oblongata, found at the base of the brainstem, is responsible for controlling involuntary functions such as heart rate, respiration, and blood pressure. It ensures that vital autonomic processes continue without conscious effort. The reticular formation, which extends throughout the brainstem, plays a significant role in arousal, sleep, muscle tone, and pain modulation. This network of neurons helps regulate states of consciousness and motor control. Cranial nerves emerge from various regions of the brainstem, carrying both sensory and motor information essential for functions such as swallowing, facial movement, and autonomic regulation. This lateral perspective of the brainstem emphasizes its crucial role in integrating sensory information, coordinating motor output, and maintaining essential autonomic functions necessary for survival. The cerebellum plays a crucial role in coordinating movement and ensuring smooth, precise motor control. It processes sensory input from multiple sources to fine-tune motor activity. Information is received from the inner ear, which provides balance and spatial orientation, as well as from visual input, allowing the cerebellum to adjust movement based on what is seen. Additionally, proprioceptors located in the muscles, tendons, and joints send sensory feedback about body position and movement. By integrating these sensory signals, the cerebellum refines and coordinates the execution of movement initiated by the motor cortex. This ensures that movements are fluid, accurate, and properly timed, preventing imbalances or uncoordinated actions. Without cerebellar coordination, motor functions such as walking, reaching, and maintaining balance would be less controlled and inefficient. The cerebellum's continuous adjustments allow for precise motor control and adaptability in movement. The diencephalon is a critical region of the brain that plays a central role in sensory processing, autonomic regulation, and endocrine control. It consists of three key structures: the thalamus, hypothalamus, and epithalamus. The thalamus acts as a relay station for sensory information. Nearly all sensory input, except for smell, is processed in the thalamus before being directed to the appropriate brain regions for further interpretation. Additionally, communication between the motor cortex and cerebellum is relayed through the thalamus, ensuring coordinated motor output. The hypothalamus serves as the control center for homeostasis, regulating essential physiological functions such as body temperature, osmolarity, and food intake. It is also responsible for governing behavioral drives, including hunger and thirst. As a major influence on both autonomic function and endocrine regulation, the hypothalamus ensures the proper balance of involuntary processes in the body. It is also a primary output center of the limbic system, meaning it plays a role in linking emotional responses to physiological changes, such as increased heart rate during stress. The epithalamus houses the pineal gland, which is responsible for the production of melatonin, a hormone that regulates sleep-wake cycles. This function is crucial in maintaining circadian rhythms and promoting restful sleep. Together, these components of the diencephalon ensure the integration of sensory, autonomic, and endocrine functions, allowing the body to maintain homeostasis and respond appropriately to internal and external stimuli. This figure provides a detailed look at the diencephalon, highlighting its key structures and functions. The thalamus serves as an integrating center and relay station for both sensory and motor information. It processes sensory input before directing it to the appropriate areas of the cerebral cortex, and also facilitates communication between the motor cortex and cerebellum. The pineal gland, located within the epithalamus, is responsible for melatonin secretion, which regulates the sleep-wake cycle and helps maintain circadian rhythms. The hypothalamus plays a crucial role in homeostasis, controlling essential functions such as body temperature, thirst, hunger, and autonomic regulation. It also influences behavioral drives, helping regulate emotions and physiological responses to stress. The pituitary gland, located just below the hypothalamus, is often referred to as the master endocrine gland. It secretes hormones that regulate growth, metabolism, reproduction, and stress response by influencing other endocrine glands in the body. Together, these components of the diencephalon ensure proper coordination between sensory processing, autonomic control, endocrine regulation, and behavioral responses, making it an essential hub for maintaining internal balance and physiological stability. The diencephalon is a central structure of the brain located between the brainstem and the cerebrum. It serves as a major hub for sensory processing, autonomic regulation, and endocrine function. This region consists of three primary components: the thalamus, hypothalamus, and pineal gland. The thalamus acts as a relay station, processing nearly all sensory information before transmitting it to the appropriate areas of the cerebral cortex. It also plays a role in motor coordination, assisting in communication between the cerebrum and cerebellum. The hypothalamus is responsible for homeostatic regulation, controlling functions such as body temperature, hunger, thirst, and autonomic responses. It also exerts control over the pituitary gland, influencing hormone secretion and endocrine system regulation. The pineal gland, located near the thalamus, produces melatonin, which regulates sleep-wake cycles and circadian rhythms. Additionally, the pituitary gland, seen at the base of the hypothalamus, is divided into the anterior and posterior pituitary lobes. It serves as the master gland, releasing hormones that regulate growth, metabolism, and reproductive functions. Together, the components of the diencephalon ensure the seamless integration of sensory, autonomic, and endocrine functions, maintaining the body's internal balance and physiological stability. The bones of the skull provide a rigid enclosure that shields the brain from injury and trauma. The lobes of the cerebrum are named after the overlying skull bones. The frontal bone corresponds to the frontal lobe, which is responsible for higher cognitive functions, decision-making, and voluntary movement. The parietal bone covers the parietal lobe, which is involved in sensory processing, spatial awareness, and touch perception. The temporal bone protects the temporal lobe, which plays a key role in hearing, language comprehension, and memory formation. The occipital bone lies over the occipital lobe, which is primarily responsible for visual processing. These skull bones form a strong protective barrier, ensuring that the brain remains secure while allowing for neural function and development. This figure presents a lateral view of the central nervous system and the brain, emphasizing the structural organization and functional regions of the cerebrum and cerebellum. In the lateral view of the central nervous system, the cerebrum is the largest and most developed part of the brain, responsible for conscious thought, voluntary movement, and sensory processing. The spinal cord, extending from the brainstem, serves as the primary communication pathway between the brain and the body. The lateral view of the brain highlights the four cerebral lobes, each associated with specific functions. The frontal lobe governs decision-making, problem-solving, and voluntary motor control. The parietal lobe processes sensory information related to touch, temperature, and body position. The temporal lobe is responsible for hearing, language comprehension, and memory formation. The occipital lobe is dedicated to visual processing. The mid-sagittal view of the brain provides a deeper look into the brain's internal structures. The cerebellum, located at the base of the brain, ensures movement coordination, balance, and fine motor control. The brainstem, which includes the midbrain, pons, and medulla oblongata, controls autonomic functions such as respiration, heart rate, and reflex actions. These structural perspectives illustrate how different brain regions work together to regulate movement, sensory processing, cognition, and autonomic functions, ensuring proper body coordination and homeostasis. The cerebrum is the largest and most complex part of the brain, responsible for higher cognitive functions such as intelligence, reasoning, planning, sensory integration, motor control, and decision-making. The cerebrum consists of two hemispheres, which contain both gray matter and white matter, and are connected by the corpus callosum, a large bundle of axons that allows communication between the left and right hemispheres. The gray matter of the cerebrum includes several key structures. The cerebral cortex is the outermost layer where conscious thought, perception, and voluntary movement control occur. The basal nuclei play a role in movement regulation, ensuring smooth and coordinated motor activity. The limbic system, which will be discussed further in an upcoming slide, includes structures like the amygdala and cingulate gyrus, which regulate emotions and memory processing. The hippocampus is also part of the limbic system and is essential for learning and memory formation. The white matter of the cerebrum is primarily found in the interior of the brain. It consists of bundles of myelinated axons that connect different regions of the brain, facilitating efficient communication between sensory, motor, and cognitive processing centers. The cerebrum's integration of sensory input, motor commands, and cognitive processing enables humans to engage in complex behaviors, problem solving, language, and emotional regulation, making it the center of higher brain function. This figure illustrates the gray matter of the cerebrum, which plays a critical role in sensory perception, motor control, decision-making, and cognition. The cerebral cortex and basal ganglia are two of the three primary regions of gray matter in the cerebrum. The third region, the limbic system, will be explored in an upcoming slide. The left portion of the figure provides a cross-sectional view of the cerebrum, revealing key structures. The basal ganglia are deep clusters of gray matter that regulate movement coordination and motor learning. The corpus callosum, composed of white matter tracts, connects the left and right hemispheres, facilitating communication between them. Surrounding the lateral ventricles, the gray matter of the cerebral cortex is responsible for higher cognitive functions and conscious thought. The right portion of the figure highlights the organization of neurons within the cerebral cortex. Neuronal cell bodies form distinct layers and columns, each specialized for processing different types of information. The outermost layers of the cortex contain gray matter, while deeper layers transition into white matter, which consists of myelinated axons that connect different brain regions. This layered structure of the cerebral cortex allows for efficient information processing, ensuring that sensory input, motor commands, and cognitive functions are integrated seamlessly for optimal brain function. The limbic system is a collection of brain structures that play a key role in emotions, memory, and learning. Anatomically, it is part of the gray matter of the cerebrum and is deeply interconnected with other brain regions that regulate behavior and autonomic functions. Three primary structures make up the limbic system: the amygdala, hippocampus, and cingulate gyrus. The amygdala is responsible for processing emotions, particularly those related to fear, aggression, and social behaviors. It plays a crucial role in the formation of emotional memories and is involved in the brain's fight or flight response. The hippocampus is essential for learning and memory formation. It helps convert short-term memories into long-term storage, making it vital for learning new information and recalling past experiences. The cingulate gyrus is involved in emotion regulation and cognitive processing. It helps link behavioral responses to emotional experiences and contributes to decision-making and attention regulation. Although the thalamus is visible in this diagram, it is not part of the limbic system. However, it serves as a relay center that processes and directs sensory information to appropriate areas, including the limbic system. By integrating emotion, memory, and cognition, the limbic system influences behavior, motivation, and decision-making, playing a critical role in both conscious thought and subconscious emotional responses. The cerebral cortex is responsible for higher-level brain functions, including perception, movement control, and cognitive processing. From a functional viewpoint, it can be divided into three main regions. Sensory areas are responsible for translating sensory input into perception, allowing for conscious awareness of stimuli such as touch, temperature, pain, and vision. These regions process incoming information from the body and environment, helping the brain interpret sensations. Motor areas of the cerebral cortex direct skeletal muscle movement, enabling voluntary control over body movements. These regions generate signals that travel through motor pathways, ultimately coordinating muscle contractions and motor responses. Association areas integrate sensory and motor information, allowing for more complex cognitive functions. They contain working memory, which helps provide meaning to sensations, linking them to past experiences. The motor association areas store memories of previously learned movements, enabling coordinated and refined motor responses. By working together, these functional areas allow the cerebral cortex to interpret sensory information, initiate movement, and integrate past experiences into decision-making and learning. The cerebral cortex is divided into functional areas that allow for sensory perception, motor control, and integration of information. These areas are distributed across the four primary lobes: the frontal, parietal, occipital, and temporal lobes. The frontal lobe houses the primary motor cortex, which controls voluntary movements. The premotor area and motor association area help refine and coordinate complex motor tasks. Additionally, the prefrontal association area is responsible for decision-making, problem-solving, and behavioral planning. The parietal lobe contains the primary somatosensory cortex, which processes sensory input related to touch, pain, temperature, vibration, itch, and proprioception, the body's awareness of its position and movement. This lobe integrates sensory information from the skin, musculoskeletal system, and internal organs. The occipital lobe is dedicated to visual processing. It contains the visual cortex, which interprets input from the eyes, allowing for the perception of color, shape, and motion. The temporal lobe plays a key role in hearing, language comprehension, and memory formation. It houses the auditory cortex, responsible for processing sound, and the olfactory cortex, which interprets smell. The gustatory cortex, involved in taste perception, is also located in this region. Additionally, a deep cortical region called the insula lies beneath the lateral sulcus and plays a role in processing visceral sensations, emotions, and autonomic functions. By coordinating sensory input, motor commands, and cognitive functions, the cerebral cortex enables perception, movement, learning, and higher-level thought processes, making it essential for daily activities and complex behaviors. The cerebral hemispheres are not functionally identical. Certain cognitive and motor functions are more dominant in either the left or right hemisphere, a concept known as cerebral lateralization. The left hemisphere is typically associated with language, verbal skills, mathematical reasoning, logic, and analytical processing. It controls the right hand and is responsible for speech production, writing, and language comprehension. The general interpretive center, which processes language and numerical calculations, is also located in this hemisphere. Additionally, the auditory cortex in the left hemisphere primarily processes sounds from the right ear, while the visual cortex processes information from the right visual field. In contrast, the right hemisphere is more specialized for visual and spatial skills, artistic ability, intuition, and creativity. It controls the left hand and is responsible for spatial visualization, facial recognition, and emotional interpretation. The auditory cortex in the right hemisphere processes sounds from the left ear, and the visual cortex processes input from the left visual field. The corpus callosum, a large bundle of nerve fibers, serves as the communication bridge between the two hemispheres, allowing for the integration of information from both sides of the brain. Although certain functions are lateralized, both hemispheres work together to enable complex thought, problem solving, and coordinated motor activity, ensuring seamless cognitive and sensory processing. Brain imaging techniques help researchers and clinicians visualize brain structure and function. In vitro techniques include horseradish peroxidase, which uses enzyme transport to trace neural pathways, rainbow mice, a genetic method where neurons fluoresce in different colors to map brain circuits, and clear lipid exchange anatomically rigid imaging compatible tissue hydrogel, a technique that makes brain tissue transparent for three-dimensional visualization of neural networks. In vivo imaging allows real-time observation of brain activity. Electroencephalography records electrical signals from neurons, useful for studying brain waves in sleep. Positron emission tomography highlights metabolically active brain regions using radioactively labeled glucose, while functional magnetic resonance imaging detects changes in blood flow and oxygen levels, providing insights into brain function during cognitive tasks. These methods are essential for understanding neural activity and diagnosing in neurological conditions. The human body follows a circadian rhythm, alternating between periods of wakefulness and sleep in a 24-hour cycle. This rhythm regulates various physiological processes, including body temperature, blood pressure, and hormone secretion, ensuring they fluctuate appropriately throughout the day. The suprachiasmatic nucleus, located in the hypothalamus, serves as the body's internal clock. It receives visual input from the optic nerve, detecting changes in light and darkness to synchronize the sleep-wake cycle with the external environment. The neuronal and hormonal activities it generates influence numerous bodily functions, helping maintain a consistent biological rhythm over a 24-hour period. Language processing in the brain is primarily controlled by two key regions, Wernicke's area and Broca's area. Wernicke's area is responsible for language comprehension, allowing individuals to understand spoken and written words. Broca's area, on the other hand, controls language expression, enabling speech production and written communication. Damage to these areas leads to aphasia, a language dysfunction. Receptive aphasia, caused by damage to Wernicke's area, results in an inability to understand language. Individuals with this condition may produce speech, but it often consists of nonsensical words or phrases. Expressive aphasia, linked to damage in Broca's area, affects speech production. Individuals can understand spoken and written words, but struggle with forming coherent sentences. They may say the correct words, but in a disorganized or grammatically incorrect order. These language centers work together to ensure fluent communication, with Wernicke's area processing input and Broca's area coordinating the motor control needed for speech and writing. Language processing involves multiple brain regions working together to comprehend and produce speech. This figure illustrates how the brain processes both written and spoken words before generating a verbal response. When reading a written word, information first travels to the visual cortex, which interprets the written symbols. This data is then sent to Wernicke's area, where it is processed for meaning and comprehension. From there, the information moves to Broca's area, which formulates the spoken response. Finally, signals are sent to the motor cortex, which controls the muscles required for speech production. When hearing a spoken word, the process begins in the auditory cortex, which interprets sound signals. These signals are then relayed to Wernicke's area for comprehension. Just as in reading, the information is transferred to Broca's area for speech planning and then sent to the motor cortex, where verbal expression is initiated. These interconnected neural pathways ensure that the brain can efficiently process both written and spoken language, enabling effective communication and response. This presentation covered the major components and functions of the nervous system, focusing on 5 key topics. We explored the evolution of nervous systems, tracing their complexity from simple neural structures to the highly developed human brain. We examined the anatomy of the central nervous system, identifying its structural organization and protective features. The spinal cord was highlighted as the main communication pathway between the brain and body, integrating sensory and motor information. Our discussion on the brain covered its regions and functions, including the cerebrum, cerebellum, brainstem, and limbic system. Finally, we explored brain function, emphasizing sensory and motor processing, language, memory, and higher cognitive abilities. Together, these topics provide a comprehensive understanding of the nervous system's role in regulating body functions, processing information, and enabling complex behaviors.